Hi guys, today we are going to be talking about chapter 23 and continuing our evolution unit. And in chapter 23 we're going to be talking about how populations evolve. And if you remember from our last chapter, populations are groups of the same species that are able to interbreed with each other. So today we're going to talk about how natural selection affects populations and causes them to evolve. So in the last chapter, we talked about what natural selection was, and that is just the environment causes organisms to adapt, and the best organisms suited for that environment are going to have more babies, and then their babies are going to have those traits that are going to make them better for the environment. So natural selection actually only acts on individuals. So individuals of a population is just one organism, one thing. But only populations evolve. So natural selection acts on individual organisms, but species as a whole evolve. There are what we call genetic variations in a population that contribute to evolution. So genetic variation is just what it sounds like, a variation in the genes, variation in different traits. So that helps with evolution. And then there's this thing called microevolution, which is a change in allele frequencies in a population over different generations over time. And if you remember from our genetics unit, alleles are just different form of a genes, and you can have dominant recessive alleles. And then there are two processes, mutations and sexual reproduction, that actually produce these variations, these changes in the gene pools that help contribute to differences in populations and individuals. So this is just an example of genetic variation. We have two caterpillars. One has these weird bumpy looking things all over it, and one is a little bit more smooth. So these are probably the same type of caterpillar, but they have different phenotypes, different things that we can see based on the variation in their genotypes, their genetics. So there are scientists that are called population geneticists, and they measure what we call polymorphisms in a population, so different forms of a phenotype of what we see in a population by determining the amount of heterozygous organisms at the molecular levels in their genes. So if you remember from our genetics unit, heterozygous or heterozygosity is basically just you are a hybrid of the dominant and the recessive trait. Okay, so population geneticists look at the amount of heterozygotes in a population. So the average heterozygosity measures the average percent of individuals that are actually heterozygous in a population. So we're not going to make it more complicated than it is. How many heterozygous hybrids are there? That's the average heterozygosity. Most species exhibit what we call geographic variation, and that's exactly what it sounds like. There are differences in populations, in species, in their gene pools because they are geographically farther apart from each other, or they're in different geographic regions. And then some examples of geographic variations occur in what we call a cline, which is basically just a change in a trait along a geographic axis. So we can actually see that there is a change, and you can see it's called graded, which means you can see the steps. Oh, this is changing because the environment is slowly changing along this axis in the geography. So this is an example of a cline, and over here we can see the allelic frequency, and then this is the latitude. So this is the frequency of the allele of the heterozygotes, and you can see that as they go further north, further south from going to cold to warm, the frequency in this fish of whatever trait this is, I don't know what this gene stands for, it goes down based on how warm it is. So, mutations, we talked a little bit about that when we talked about the DNA unit, and mutations are just changes in DNA. That's all it is. So you change the nucleotides, the letters, adenine, thymine, cytosine, guanine. Any change in DNA is called a mutation. And mutations can actually cause new genes and alleles to form. So new genes, different forms of a gene to form, because we have different amino acids being made, different proteins being made because of these mutations. And then only mutations in cells that produce sex cells, gametes, can be passed to offspring. Well, that makes sense because only the sperm and the eggs create an offspring, create a zygote. So only mutations in those cells are actually passed to the babies. And then a point mutation is a change just on one base pair, one A, T, C, or G in a gene. So point mutations, the effects of them and the effects of that change can be different. They can be not harmful, they can be harmful. So these are the effects of point mutations. So mutations in non-coding regions of DNA are harmless. And if you remember from our DNA unit, non-coding regions of DNA are just pieces of DNA where they don't get read. They're just kind of there. No proteins are made from them. So mutations on this part, they're harmless because nothing's being made from them. They're just kind of sitting there. Mutations in a gene it probably won't affect protein production because there is redundancy in the genetic code. And remember, in our nucleotides and our nitrogenous bases, there's a lot of the same thing over and over and over. So even if one of them gets messed up, well, there's something there to fix it right after that. But mutations that result in a change in proteins are pretty much harmful, because you can cause different proteins to be made where they're not supposed to be, or you can stop proteins from being made. So pretty much changing DNA wherever proteins are going to be made is bad most of the time. But mutations that result in a change in a protein that is actually a benefit can actually increase the fitness of an organism to its environment. Most of the time, mutations aren't that great. At best, they're not going to hurt you. But sometimes mutations are good because they make you more fit for your environment. So chromosomal mutations is just what it sounds like, mutations on your chromosomes. They can either delete, disrupt, or rearrange genes in loci or just locations of genes that are typically harmful. So mutation rates are low in animals and plants. We don't see mutations happen very, very often, but they are definitely a lot higher in prokaryotes and even higher in viruses. So you can see from our last chapter when we talked about how HIV viruses are also always changing and adapting really, really quickly. I mean, we can see that from generation to generation. But in animals and plants, it takes forever to see mutations change a population. Sexual reproduction is another thing that affects evolution and changing in a species. And sexual reproduction, we all know what that is. That's reproduction, making new organisms involving two organisms, a sperm and an egg. And sexual reproduction can actually shuffle existing alleles into new combinations. When we talked about a few units ago, when we talked about meiosis, we talked about the chromosomes actually exchanging DNA with each other and how the chromosomes line up, producing many, many, many possible combinations of chromosomes in the baby. So this is what we're talking about again. When organisms reproduce sexually, the alleles combining and shuffling up is more important than mutations, than producing genetic differences. And this is what makes adaptation possible, is the shuffling of genes. So, I'm going to talk about this thing called the Hardy-Weinberg equation. It's going to be a little bit difficult for me since I'm not in front of you to go over it, so I will post some more videos of people doing problems to help you with it. But the Hardy-Weinberg equation basically tells you whether a population is evolving currently or if they're being stable. Okay, so we're going to go over this equation in a couple slides. So we talked about this already, but a population is a species in an area. Okay, same type of organism that are able to reproduce with each other, and their offspring are fertile. So if you think about a horse and a donkey, they can mate and they can have babies, but their offspring mules, they are not fertile, so they are not the same. Same thing with a lion and a tiger. Well, you can get a liger or a ligon, but their offspring aren't fertile. They're not the same species. So a population is a group of organisms that can have babies A gene pool consists of all of the alleles for all of the organisms in a population. So if you look at the gene pool in the classroom, it's all of the genes for all of the alleles in the classroom. That's our gene pool. If you look at the human population as your gene pool, it's all of the organisms, all of their genes, all of their alleles for the whole entire population. That's what a gene pool is. A locus is fixed if all individuals in a population are homozygous for the same allele. So basically, if we're homozygous dominant or recessive, if everyone has the same exact trait, if we're all homozygous for dominant, let's say dark hair color, then we're not evolving because everyone's going to have a dark hair, and our babies are going to have dark hair, their babies are going to have dark hair. So evolution really on populations depends on the differences in whether you're heterozygous, homozygous, dominant, and recessive. So here is the Hardy-Weinberg equation. So the frequency of how often an allele shows up, so how often dominant or recessive shows up in a population, can actually be calculated based on this equation right here. So if there are two alleles that represent a gene at a locus, at a specific location, the letters P and the letter Q are used to represent how often they show up. Okay? So the frequency of all the alleles in a population should add up to 1. So we have P plus Q equals 1. So the Hardy-Weinberg principle, P plus Q equals 1, that describes an allele, a population that is not evolving. So the closer a population is to this P plus Q plus 1, to the Hardy-Weinberg principle, the more stable it is. Okay, so if we're not evolving, we're definitely going to be more stable. If all of us have that dark hair, and we're looking at the trait for dark hair, the alleles for dark hair, then we're going to be at P plus Q plus 1, okay? We're going to be at that Hardy-Weinberg principle. But to actually calculate genotypic frequencies, or how often genotypes show up, we have to do some more work to this equation. So the equation is P squared plus 2PQ plus Q squared equals 1. And that looks all kinds of crazy, but we're going to explain what that is. So P squared and Q squared represent the frequencies, or how often homozygous genotypes are. So let's just say we got P squared, that's homozygous dominant, if you want to think of it like that. Q squared is homozygous recessive, and then 2PQ, this part right here, is how often the heterozygous genotype shows up. So P squared is homozygous dominant, if that makes it easier for you. 2PQ is how often heterozygous shows up. And then Q squared is how often homozygous recessive shows up. So there are 5 conditions for populations that are not evolving. Okay, so we got a stable population. They have to meet 5 requirements. They're not evolving. There can be no mutations. So in a population that's not evolving, there's no mutations. In a population that's not evolving, there is no random mating. And in a population that's not evolving, there's no natural selection. It has to be a big population, and there is no gene flow or movement. Now, you can see from this bullet right here, well, that is probably really never going to happen that often in nature, because all of these things just happen in nature. That's why evolution and adaptation is just so common, because these 5 conditions are rarely ever met. So, when we're looking at this Hardy-Weinberg principle, here's an example of a locus that causes PKU, and which is basically just a disorder. So PKU, the gene mutation rate is low. Okay. Mate selection is random with respect to whether or not an individual is a carrier of the PKU allele. Okay. Natural selection can only act on rare homozygous individuals who do not follow these dietary restrictions. The population is large. Migration has no effect on the allelic frequencies. So we're looking at those 5 things that we talked about, those 5 conditions for this PKU disease, and so we're going to see if we can apply the principle to that. So this disease is the occurrence of PKU is 1 per 10,000 births. So our Q squared, which we talked about was the homozygous, okay, equals 0.001. And then if you want to take away this squared part, we're going to move over our decimal point, and Q equals 0.01. And we got that number from 1 divided by 10,000, the 0.0001. The frequency of the normal allele, okay, therefore, would be 0.99. Okay, because remember we got P plus Q equals 1. So if Q is 0.01, well, 1 minus 0.01 is 0.99. Okay, and then that the frequency of heterozygotes, so the carriers, is 2PQ. So we have 2 times 0.99, because this is where we figured out where 0.99 was, and we multiply that by 0.01. So, and with that, when you multiply all of these together, we get 0.0198. So, approximately 2% of the United States population has this disorder, PKU. Okay? So, PKU happens 1 in 10,000 births. 1 divided by 10,000 equals 0.0001. We want to take away that squared, so we move our decimal point over 2, and that means that Q equals 0.01. So if we take the equation from a couple slides ago, P plus Q equals 1, and we subtract 0.01, because Q equals 0.01, we get 0.99. So P equals 0.99, and then we just plug in that 0.99, 0.01, and then we multiply it by 2. And I know that's a little bit confusing. I really wish I could figure out how to write on my screen, but I will post some videos to help you figure this out. So we're going to move on to talking about genetic drift in gene flow and how it relates to natural selection. So there are 3 major factors that alter how often alleles show up, and then how often this brings about change in evolution. So we have natural selection. We talked about what natural selection is. I really hope you know what it is by now, but that is the environment, you adapting to your environment and changing a population. So natural selection is non-random because you are adapting to things that are happening in your environment. We have genetic drift, which is random, and we have gene flow, which is random. So we're going to talk about each of those. So natural selection is basically success in reproduction. Okay, so if you are successful in your environment, it's going to bring about certain alleles to the next generation. So if you're more fit, obviously you're going to have more babies. Your babies are going to have the traits that you have and change the population that way. Genetic drift describes how often dominant and recessive alleles show up and how they fluctuate randomly from one generation to the next. So, how often dark-haired people versus light-haired people show up is random, and it's going to change every generation. So that's just an example of what genetic drift is. The smaller the sample size, though, the greater the chance of deviation from a predicted result. So we have to look at as big of a population size as possible to get really good information. But because small sample sizes don't really show what we expect, because obviously more organisms you look at, more variations in traits. And genetic drift reduces genetic variation through losses of dominant or recessive alleles. So this is just an example of genetic drift, okay? So we have the first generation here. The P in our Hardy-Weinberg equilibrium equation was 0.7. So in the Q, which was 0.3. So we have a really good mixture here of genetic drift, and then you can see it's 50-50. In the second one, the dominant and the recessive are 50-50, and then eventually the third generation, based on how often these traits showed up, ended up with all red flowers. So in genetic drift, there's this thing called the founder effect. And the founder effect is basically when a couple organisms actually become isolated from the rest of the population. So we have a big population, there's some organisms that kind of got thrown over here and they're all by themselves for whatever reason, due to like natural disasters or just happening to migrate away, that kind of a thing. It can affect evolution and it can change a population. So how often dominant and recessive alleles show up in this small founder population, the population that kind of went off on its own, the frequencies can actually be different from those in the larger parent population. Now that makes sense because if only a couple of organisms went this way or got separated from the group, well, there's only a couple of traits that are going to show up versus a huge population. So in genetic drift, there's another thing called the bottleneck effect. And what that is when there's a sudden reduction in population size. So let's say, I don't know, a giant fire comes in and wipes out most of whatever species there is. Now that's called the bottleneck effect. So if the majority of a species is wiped out, well, there's only a few left. So the direction of how the population is going to look is going to be a result. It's going to change based on the only the few that are left after the natural disaster. So the gene pool is going to be a lot smaller. And if the gene pool remains small, it probably will be affected by genetic drift. So here's an example of the bottleneck effect. Let's pretend all of these little marble-looking things in there are organisms. So you can see that the original population in the quote-unquote bottle we have blue and white and yellow. Now, for some reason, we have a natural disaster like an earthquake or a tornado or a fire. Well, only a few of those organisms escape and they make it out. So the surviving population is not going to have as much variation. So there's not even any yellow marbles in here, so that's probably not going to show up. And there's more blue than white when before they were equal. So the population of these marbles are going to most likely end up being blue because that's what survived after the natural disaster. So genetic drift is really significant when the populations are smaller. Genetic drift can cause how often alleles show up, the dominant and the recessive, to change at random. It can cause organisms to lose genetic variation, and it can be harmful if alleles become fixed and there's not a variation of genes. So gene flow is another thing that can cause evolution. Gene flow is exactly what it sounds like. It's moving genes. So gene flow can either be from emigration or immigration. So immigration is moving into a population, emigration is moving out. So alleles can be transferred from when organisms that are fertile, that can have babies, move in or out of a gene pool. So gene flow actually can reduce differences in populations over time, and it can actually, gene flow is more likely than a mutation to change allele frequencies directly. So gene flow can, because you have people or other organisms coming into a population or out of a population, it can change what we look like. So an example, and this is probably the biggest example that I can think of for gene flow, is people. So we have different populations of people that occur naturally, and we just look different because of we adapted to our environments. People that are closer to the equator are going to be darker because their skin has to protect them and fill with melanin, kind of like tinting car windows to protect from damaging UV rays. And people who live farther from the equator tend to be lighter. So gene flow in these different populations causes us to look different. So when a lighter person and a darker person, when they enter each other's quote unquote populations and then they have babies, well, their babies are going to be, they're going to be a mixture of the two. So that's an example of gene flow in the human populations, because even in different geographical areas we look different, but if we mix the people from those different geographical areas, we get people who look totally different, a mixture of the two populations. And this happens in all organisms, not just people. So right now is going to be a stopping point for today. I need you to make sure that you are working on your chapter summaries and keeping up with that. Rewatching the video if you have any questions. I will post some videos of Hardy-Weinberg equilibrium problems being done. I'm going to continue the video at this point, but today is the stopping point for today. Okay, so we're going to move on to concept 23.4, and we're going to talk some more about natural selection. So natural selection is pretty much the only thing that results in adaptive evolution. So evolution caused from adapting to your environment. And natural selection brings about these changes to the populations by acting on phenotypes. So natural selection acts on phenotypes. It acts on things that we can see or talk about. So when we talk about natural selection in the last chapter, we mentioned survival of the fittest, or the struggle for existence, but it's a little bit more detailed than that. So reproductive success, or how many babies you have, is actually a little bit more complicated than just survival of the fittest. It depends on many factors. So there's this thing called relative fitness, which is basically how much you contribute to a gene pool of the next generation, okay, relative to the contributions of other individuals in that population. So the higher your relative fitness, the more babies you're going to have, basically. Your genes are going to be thrown into that gene pool more than in other individuals. So natural selection actually favors certain genotypes by acting on the phenotypes of these organisms. So there are 3 different types of natural selection. We have directional selection, disruptive, and stabilizing selection. So directional selection favors individuals that are at one end of the phenotypic range. Disruptive selection favors individuals at both ends of the phenotype. And then stabilizing selection favors phenotypes that are in the middle and acts against extreme phenotypes. So here's an example. So we have these mice, Here's the original population. Now, these guys in the middle, okay, those are those are the intermediate. Those are the middle guys, okay? And then the guys at the end are going to be the extremes of the phenotype. And obviously the phenotype is fur color in this example. So directional selection, which we talked about on the last slide, favors an individual at one end of the spectrum. So for some reason, these mice over here, it is better for them to be in that environment and be darker fur color. In disruptive selection, which is in the middle, okay, we talked about on the last slide, it favors individuals that are at the extreme ends of the phenotypic range. So for some reason, in this type of natural selection, it was bad for these mice to be in this middle color. Maybe the predators could see them more or something like that. So in disruptive selection, it's better for the mice to be white or this darker gray color. And then stabilizing selection, it's better for the mice in this example to be this middle color here, and then the extreme ends of the spectrum, the light and the dark, both got wiped out for some reason. So natural selection is really important. We've established that. We talk about it on almost every other slide in this unit. But natural selection increases the frequencies of the dominant and recessive alleles that actually enhance how well you survive and how often you reproduce. Okay? Adaptive evolution is basically this match or this standoff between an organism and how well it lives in its environment. So here are a couple examples of adaptive evolution. So the example up here, there's actually a little thing in here called a cuttlefish, and they're really cool if you want to go look at YouTube videos of them. But they've actually been able to adapt to their environment by camouflaging themselves, okay? And then snakes have actually adapted the ability to unhinge their jaws to move them so that they can swallow their large prey. So, environment in the earth, always changing. Everything is always changing. Evolution, therefore, has to always be changing as well. So, organisms are always adapting to their environment. It's a continuous process. The environment is always going to change no matter what, and organisms either have to adapt with it or they have to die off. That's all it is. So, genetic drift and gene flow are random, and they don't really lead to adaptive evolution because these are things that are happening. Boom, natural disaster happens, so we get the bottleneck effect. They are random. It has nothing to do with the gradual change of the earth. Natural selection is also going to gradually change with the changes in the environment. We're going to switch gears a little bit and talk about sexual selection. And this is basically another thing that causes evolution by natural selection. And sexual selection is a type of natural selection for mating. And basically what that is organisms choose traits that they find favorable when they're looking for a mate. Okay? So, like the guppy example in the last chapter, the female guppies like the fancier male guppies, so they wanted to mate with them. So that changed the population. That's what sexual selection is. Now, sexual selection can result in this thing called sexual dimorphism, which is basically differences between the sexes in their characteristics. So this would be like peacocks being super brightly colored, and the peahens, the females, they're drab, they're dull, and like our guppy example that we just talked about. So sexual dimorphism is just when the males and the females look different, like vastly different. Male showiness, or the males being more brightly colored due to mating choices, actually will increase the chances of attracting a female, but it decreases its chance of survival. So the more brightly colored you are, the more attention you bring to yourself. Oh, so I jumped ahead of myself with the example, but this is just the peacock example that I showed you before, sexual dimorphism. On the right is the peacock, which is the male, and then here's the peahen. She's more boring looking, which is sad for her, but she is more dull colored. Now, obviously, if a peacock is doing this, he's going to attract the ladies, but he's also saying, hey, here I am, come eat me. So that's why it can be bad for him as well as advantageous for him to be this showy. So female preferences actually can evolve, they can change based on what the environment is saying or based on what is good at that time. So there's the good genes hypothesis suggests that if a trait is related to male health, okay, both the male and the female preference should be selected for. Okay, so genetic variation, we talked about that, is that how the genes vary in a population, different traits, different genetics. Genetic variation is really important, and we have to try and preserve it. And there are various different things that actually help preserve the variation in a population. Because if you have too many of the same genes, that's not good for the population. It makes it less stable. So there's this thing called diploidy or diploidy, which helps maintain genetic variation with recessive alleles. So you may think that a population, hey, we all got brown hair, but then there's someone there that is hiding those recessive alleles. So that's what diploidy is. There's another thing called heterozygote advantage, where it's basically it's advantageous. It's good for you to be a heterozygote because you have a higher fitness than the ones that are either homozygous dominant or recessive. So an example would be sickle cell disease. Okay, so sickle cell disease, obviously it's really bad if you have sickle cell disease. So you're homozygous for sickle cell, you have the disorder. If you're homozygous to not have it, then you're just normal or whatever. But heterozygotes, to have the advantage, they're actually resistant to malaria. And that's a big deal because in Africa, where there's a huge malaria is huge in Africa, the people who are heterozygous, so they are a carrier for the sickle cell gene, but they're not sick, they're actually resistant to malaria, and they tend to live in areas that are infested with mosquitoes that carry malaria. So it is good for them to be heterozygous for one affected gene and one unaffected gene. So, frequency dependent selection is basically the fitness of a phenotype going down if it becomes too common. So if we have a phenotype that is just way too common, something that we see that's way too common, it actually makes your fitness go down in that population. So natural selection actually favors the phenotypes that are less common. Neutral variation is genetic variation that doesn't really have any advantage or disadvantage. So neutral variation is the change up in our non-coding regions of DNA. So it doesn't matter if there's variation there because our DNA is not making proteins. And then variation in proteins that don't really have an effect on our reproductive fitness is another example of neutral variation. So if you probably asked yourself, well, if natural selection is always happening, why is it that organisms still aren't like this awesome, perfect thing? Well, natural selection can only act on existing variations. Evolution is limited by things in the environment, okay, things that have happened in history. Adaptations are actually compromises. So they're actually compromises one thing for the other. And then chance, natural selection, and the environment always interact with each other. So it's not perfect. You may be better for your environment, but no organism is ever going to be perfect because the environment is always changing. So that is the end of this chapter. You should now be able to explain why a lot of point mutations are harmless. Explain how sexual recombination generates different variability, different variation in genes. You need to be able to tell me what population, species, gene pool, relative fitness, and neutral variation are. You have to know about Hardy-Weinberg equilibrium, and it is important that you know the 5 conditions of organisms being in equilibrium. Okay, you have to be able to do this equation, and like I said, I will post some more example videos. And you have to explain why natural selection is the only mechanism that consistently produces adaptive change. Explain how population size affects genetic drift. You have to be able to distinguish the differences between directional, disruptive, and stabilizing selection, and then different types of sexual selection, and then list 4 reasons why natural selection can't produce perfect organisms. So, if you have any questions, please rewatch any part of the video that you need to. Send me an email. Go look for resources online. Read your book. Please make sure that you are keeping up with your chapter summaries, studying for your tests. And like I said, if you have any questions, just email me.